In this lesson, we'll take a look at oogenesis. So basically, oogenesis is the formation of the female gametes, which we're going to call the oocyte. This is what common laymen call the egg, right? But we're going to call it the oocyte. Now, this process actually begins very early in a female's development. You can see here it actually starts during fetal development. We have these cells called germ cells, primordial germ cells. They're in the yolk sac and they migrate to where the ovaries are going to be. And these primordial germ cells differentiate into cells known as oogonia. Now, the oogonia, they further develop into something known, what I highlighted right here, as the primary oocyte. These cells are going to enter a type of cell division known as meiosis. Right? Meiosis has two stages. There's meiosis 1 and meiosis 2. Well, they only are going to go into meiosis 1, but they don't go through the whole process. They're actually going to enter prophase of meiosis 1 and then they stop. They're going to stay suspended in this state until the female turns, or I would say what age, but goes into puberty, because again, it's variable from female to female what age they go into puberty. So these primary oocytes begin the process of meiosis. Now, I just want to remind you, if you watched the lesson on spermatogenesis, we talked about spermatogenesis. The sperm needs to also go through meiosis because it only has to have 23 chromosomes, and that's why the oocyte also will go through meiosis. It only wants to have 23 chromosomes. It doesn't want to have 46. Okay, so the concept here is we have the primary oocyte. It's going to go into meiosis, but it's only going to start it and not complete it. Right? And it's going to stay that way until puberty. So the primary oocyte is actually encapsulated, right? It's just not sitting there. It's encapsulated by what we call a primordial follicle. Right? One of the ways we can identify the primordial follicle is that it has a single layer of cells around it known as follicular cells. So here we can see this is where the primary oocyte would be, like right here. And then if you look closely, there's a single layer of cells around the follicle. These would be the follicle cells or the follicular cells. It's the same thing. Okay, so now we have the primary oocyte living in this little capsule, if you were, known as the primordial follicle. Okay, so check this out. This is interesting. At birth, there are between 200,000 and 2 million primary oocytes in each ovary. A lot of those you're going to see disappear. So look at this. By the time the female goes into puberty, there's about 40,000 left. And over the time of a female's reproductive life, approximately 400 will mature and ovulate. Okay, so we go from having many of these down to this is the actual about the actual number that will actually reach ovulation, which we'll talk about in a moment. Okay, so we followed the female's life up until puberty. What happens at the time of puberty? Right? At puberty, all the way through menopause, each month a few primordial follicles, which we just looked at, start to grow and develop into primary follicles. These follicles, they change a little bit. Instead of having one layer of follicle cells, we start to see multiple or several layers, and we change the name of the cells to granulosa cells. Right? So if you were able to, if you were to look under the microscope, you would see the oocyte, and then you would see in the follicle multiple cells. I'll show you a picture. As the primary follicle grows, we start to see a clear area known as the zona pellucida. And then also there are some cells that develop outside the follicle known as the thecal cells or the theca folliculi. All right, so again, these are some of the things that you want to look for. If you look under the microscope with the primary follicle, you'll see the oocyte, you'll see the granulosa cells. Again, that's more than one layer of cells, the zona pellucida, and then outside that is the theca folliculi. All right, so when you take a look over here, here's where the oocyte's going to be. The granulosa cells we could see over here on this kind of clear line going this way. That I know they have it labeled over here. This is another one. This would be the zona pellucida, and then you can see outside the granulosa cells. These are going to be the thecal cells. So these are some of the things that'll clue you in that you're looking at a primary follicle. So the primary follicles are going to turn into a secondary follicle. One of the features you see with the secondary follicle is you start to see some water developing around the oocyte. All right, it may not be like one pocket of water, but eventually it will form a pocket of water that we call the antrum. So the secondary follicle, one of the features I usually look for is we start to see some water developing in between the cells. So see this little white area here? Some of this is going to be pockets of water. But otherwise, everything is the same. Zona pellucida is there, your oocyte, the granulosa cells. We're just introducing some waters. Some water. Now, the secondary follicle is now going to progress into what we call a mature follicle. Also, you may hear the term Graafian follicle. With the Graafian follicle, you'll see that antrum I was talking about. Take a look at the water now. You have this big pocket of water. It's just a clear fluid. It's very easy to identify under the microscope. That's going to be what we're now looking at as a mature follicle. So just before ovulation, the primary oocyte, remember that it started meiosis, but it never finished it. It is now going to complete meiosis, producing two cells that are half the genetic material, known as haploid cells. One is normal size, and the little one is going to be called a polar body, right? We're actually going to discard that one. That's actually going to be discarded. It doesn't become anything. The largest cell receives most of the cytoplasm, and we're going to now change it from a primary oocyte, right? Not follicle, primary oocyte, to a secondary oocyte, and that's living in the mature follicle. This is interesting also. That secondary oocyte is now going to begin the second stage of meiosis, but doesn't complete it. It actually goes into metaphase. Okay, so when ovulation does happen, it's the secondary oocyte that's going to be ovulated in a suspended state of meiosis too. This meiosis too will continue only if sperm are present and fertilization takes place. Then it will complete the process of meiosis. So that doesn't happen unless fertilization will take place. Okay, so what are we looking at? The mature Graafian follicle is now going to have the secondary oocyte. One of the things we start to see around the oocyte here and between the antrum, there are cells around the oocyte known as the corona radiata. They're going to be ovulated with the oocyte, and what's interesting, the sperm is going to have to break through these corona radiata cells. The acrosome on the sperm head has enzymes that will help to break these cells away. So the corona radiata are these cells around the oocyte. And again, one of the major features here, guys, is the antrum. Right, otherwise, everything else is kind of the same. You can see the zona pellucida, the stroma, all that's pretty much the same. The thecal cells, I should say. Okay, so in the female reproductive cycle, roughly around day 14 in a 28-day cycle, we have ovulation. If you remember from the endocrine system, there's a spike in luteinizing hormone from the anterior pituitary that's going to cause ovulation. So at ovulation, the secondary oocyte and the first polar body are going to be expelled into the pelvic cavity, right? They should make their way into the fallopian tube. So there's two options. If fertilization does not occur, right, no sperm are there, the secondary oocyte is going to degenerate, and that polar body also degenerates, and they're just going to be released from the body during menstruation. If sperm are present and one does penetrate the oocyte, it's got to break through that corona radiata, meiosis too will continue. and the secondary oocyte splits into two haploid cells, again of unequal size. The larger cell is the important one. That's what we now call the mature egg or the ovum. And the new cell that's formed between the union of uh, the ovum and the sperm is now called a zygote. All right, so this is what we're going to call a zygote. Okay, so these are the steps of oogenesis. Again, and what we walked away with was a cell, a gamete, with half the genetic material.